Hola, es un placer daros la bienvenida a esta tercera edición del ACE Global Research Summit. Desde 1995, en ACE lideramos iniciativas que conectan innovación y sostenibilidad, trabajando en equipo para transformar el futuro de la salud y el bienestar global.
En la actualidad, somos una institución reconocida a nivel internacional, respaldada por un equipo interdisciplinario, multidisciplinario, de más de 100 profesionales.
Desde el principio, nuestro compromiso ha sido firme: transformar vidas a través del conocimiento y la innovación. Hoy hemos realizado más de 32.000 diagnósticos, más de 150 ensayos clínicos y tenemos más de 22.000 muestras biológicas para la investigación.
Esto nos ha permitido que en un solo año, el 2024, hemos publicado 60 artículos científicos en revistas indexadas de primer decil y primer cuartil. Estos logros representan décadas de esfuerzo en conjunto con la comunidad científica internacional y una dedicación constante para mejorar la vida de los pacientes y sus familias.
Y todo esto no sería posible sin un gran equipo, formado por personas excepcionales que encarnan nuestros valores. Por eso, en esta nueva edición del Global Research Summit, queremos celebrar estos avances y compartir los descubrimientos que están moldeando el futuro de la neurociencia, la práctica clínica y la investigación.
Bienvenidos al ACE Global Research Summit. Bienvenidos al futuro de la investigación.
Good afternoon and welcome everyone. I'm Dr. Marta Marquia, neurologist and head of the Clinical Research and Medical Psychology program at Ace Alzheimer Center Barcelona. As the chair of this session, it is my pleasure to introduce the third edition of the Global Research Summit organized by Ace. The topic of this afternoon session is the landscape of neuropathology in Alzheimer's disease. Necropsy studies plays a pivotal role in understanding the complex mysteries of dementia, delving into the microscopic landscape of brain and offering invaluable insights into the structural and biochemical alterations underlying these conditions. By examining postmortem brain tissue, neuropathologists have uncovered many hallmark features of neurodegenerative diseases, providing crucial clues for clinicians and the whole scientific community. Moreover, neuropathologists can see features that we clinicians cannot yet detect in vivo in our patients, such as TDP-43 inclusions, Lewy bodies, ARTAC, or hippocampal sclerosis. To me, it is always exciting and also surprising to review the autopsy reports of our patients from the memory clinic, which usually contain 4, 5, or even 6 different neuropathological findings, and compare those with their clinical diagnosis, usually limited to 1 or 2 entities. Neuropathology aids in increasing our understanding of the mechanisms driving neurodegeneration, refining diagnostic criteria, and also driving the development of targeted therapeutic interventions. This afternoon, we are very lucky to have a panel of distinguished speakers who are experts in the field of neuropathology of Alzheimer's disease. Before introducing our speakers, I'd like to remind the audience that they can send their questions in the chat during the talks, and we will address them after. Our first speaker today is Dr. Margaret Flanagan. Dr. Flanagan is a neuropathologist working in clinical neuropathology and neurodegenerative research. She is the co-director of the South Texas ADRC Neuropathology Core and BICS Brain Bank, the director of the NAN Study on Aging and AD, Associated Professor of Pathology at UT Health San Antonio, and endowed Baptist Health Foundation of San Antonio's Distinguished Chair in Alzheimer's and Neurodegenerative Diseases. The goal of her research is to identify the pathophysiologic mechanisms underlying dementia to enable future development of diagnostic and therapeutic interventions. She participated in the international working group that presented the consensus-based guidelines for diagnosis of the staging of late neuropathological changes. Her lab utilizes multidisciplinary approaches to delineate the molecular mechanisms of dementia. She is also working towards developing feasible biomarkers for detecting preclinical neurodegeneration. Dr. Flanagan, the floor is yours.
Thank you for the kind introduction, Marta, and thanks for the opportunity to present today. Today, I will be talking about digital neuropathology and human brain banking, building bridges for understanding neurodegeneration. So neuropathology is a subspecialty in medicine, and this quote summarizes it quite nicely. So a neuropathologist is a physician expert in the diagnosis of diseases of the nervous system and skeletal muscles, and we function as consultants primarily to neurologists and neurosurgeons. Neuropathologists are knowledgeable in the infirmities of humans as they affect the nervous and neuromuscular systems, be they degenerative, infectious, metabolic, immunologic, neoplastic, vascular, or physical in nature. So what is brain banking? So brain banking is the practice of collecting, storing, and distributing clinically and neuropathologically characterized human brain tissue samples preserved in different ways, both fixed in formalin or paraformaldehyde, as well as frozen. And brain banking requires careful consent, ethical oversight, and standardized procedures. And as I said, we preserve the brain samples different ways in order to be able to perform different types of studies, whether it's just traditional immunohistochemical staining, biochemistry, or more complex assays. So why bank human brain tissue? So post-mortem human brain tissue is what actually provides the definitive diagnosis in many settings, as brain autopsy remains the gold standard for diagnosing a lot of the neurodegenerative diseases up to this day, even though biomarkers are being developed rapidly. It also enables research to be performed on actual human disease biology, not just models, and it enables comparisons to be performed across different diseases, age groups, and comparing disease with control brains. And brain banking to date has had a large impact on dementia research, and brain banks have been essential for our field's understanding of Alzheimer's disease, frontotemporal dementias, and other neurodegenerative conditions. The discovery of amyloid beta plaques, tau tangles, and TDP-43 pathology all actually came from brain autopsy tissue studies, and a lot of this work has enabled the development of diagnostic criteria and biomarkers that can be used during life using CSF or blood samples, as well as PET imaging. Additionally, brain banking powers longitudinal studies, and studies such as the Nun study and Honolulu Asia Aging study used brain donation to correlate the brain pathologies with the decades of cognitive, behavioral, and lifestyle data that was available from during life. And these cohorts, combined with the fact that they have brain banking as part of it as an endpoint, have revealed key insights about cognitive resilience, early changes in the brain, and aging. So brain banking combined with digital pathology is truly the future of research. And why is this? This is because you can actually combine digital slide imaging with large, well-annotated brain bank data sets, and this enables scalable and AI-driven analyses and really helps support multi-layered data. So, for example, I've just included this figure here that shows how the brain bank repository, when it becomes digitized, can really tie everything together, including the clinical data points from during life, as well as experimental data that's collected from the brain autopsy tissues themselves, such as spatial omics data, sequencing data, etc. And tying this all together really facilitates sharing and collaboration across diseases, including dementia, movement disorders, epilepsy, and other rare neurologic disorders. So the learning objectives focusing on digital pathology specifically include understanding how digital pathology can be leveraged to address some challenges that are specific to non-neoplastic neuropathology. Then I'll review how we have worked to enhance and leverage the digital slide archive infrastructure to develop the new Brain Digital Slide Archive, which is optimized for, again, non-neoplastic neuropathology samples. And then I'll wrap up with describing our collaboration between two separate U24s to expand and develop digital pathology nationally and internationally. So there are many different challenges involved when working with non-neoplastic brain tissue. So specifically, a lot of the digital pathology tools and softwares that have been specifically developed for brain tumors. Whereas in non-neoplastic brain samples, we don't have as dense of a cell population. And so a lot of the already available off-the-shelf digital pathology tools are not necessarily able to be applied to our sample type. And in neuropathology, the neuropathologic exam of the brain at time of brain autopsy remains the diagnostic gold standard in the field of neurodegeneration specifically. And in addition to this, we do semi-quantitative scoring in neurodegenerative disease research, which includes, for example, quantifying the amount of diffuse plaques that are present, or neuritic plaques or tau tangles. And I've just shown an example here of the neuritic plaques, just showing how we give scores of 1 plus, 2 plus, or 3 plus for whatever it is of interest that we're quantifying. And as I'm sure many realize firsthand, there can be significant inter and intra-rater variability when performing these assessments. And in addition to that, it's quite time consuming for neuropathologists. Traditionally, prior to COVID, digital pathology hadn't really expanded nationally, but more recently it has rapidly developed, largely due to COVID as a silver lining, I guess, of the pandemic for our field. And it's great because prior to having digital pathology infrastructure, brain autopsy histologic data was not easily shared. And lastly, there is substantial variability to take into consideration in sample preparation when generating microscopic slides, such as differences in fixation, variability in sampling, even just slight differences, such as sampling the hippocampus more anteriorly versus posteriorly, and of course, differences in tissue processing, antibodies, and protocols in different laboratories. And as you can imagine, these variations in practice can at times cause challenges when trying to harmonize data for either a multi-site research project or perhaps for for diagnostic purposes specifically. So addressing these challenges, so we aim to overcome these challenges by digitizing whole slide images in our Alzheimer Center and other brain banks to facilitate easier sharing, in addition to developing machine learning analytics and improving standardization and diagnostic accuracy across sites. And at the time, although digital pathology software packages did exist, such as QPath, which is open source, and Indica Labs makes Halo software, which these are just examples, not endorsements, we felt that the field of Alzheimer's disease and related dementia research would actually benefit the most from having a single harmonized collaborative platform across multiple institutions. However, we wanted to take our prior experience using these other softwares to both learn from our experiences when coming up with the harmonized collaborative platform, in addition to integrating it with our system to be able to take, for example, annotations that had already been performed and import them into our harmonized platform. So now to talk a little bit about the DSA and enhancing and leveraging the infrastructure for the BDSA. So the DSA, or Digital Slide Archive, had been created about 8 years ago, and it was specifically created to support large whole slide image data sets that were focused on cancer diagnoses. And this was led by the team shown here, Dr. David Gutman at Emory, Dr. Manti at Kitware, and Lee Cooper. And the DSA at the time was able to support web-based visualizations, including pathology and radiology image annotations, both ones that were traced by a human or ones that were able to be designed as apps to, for example, differentiate gray and white matter. It had been powering the International Skin Imaging Collaboration Archive with over 70,000 dermoscopic images. And this specifically, this portion was developed by Dr. Gutman with Dr. Halpern at Memorial Sloan Kettering Cancer Center. And in addition, it was used by the National Cancer Institute Surveillance Epidemiology and End Results program to enable the de-identification and easy sharing of whole slide images in the field of cancer. And it was deployed at 7 U.S. cancer centers with expansion ongoing. Then the team at Kitware, Emory, and Northwestern received an SBIR Phase 2 grant, which enabled them to further develop tools supporting bulk image de-identification of whole slide images. So we wanted to take this very comprehensive platform that had been optimized for cancer diagnosis and leverage it so that we didn't have to reinvent the wheel, so that we could spend more of our energy on harmonization and the development of quantitative tools for neurodegenerative disease research. So when enhancing and leveraging the DSA infrastructure for BDSA, we thought it was a fantastic opportunity. As I said, it was able to support multi-slide scanning vendors and file types as well, which was very helpful as many different brain banks had different slide scanners, different brands such as Olympus or Aperio that were generating different file types, which for even some of the commercially available softwares were not compatible. And the DSA provides an provides application programming interfaces in a vendor agnostic manner to be able to access and view image data, create and query specific annotations, and deploy image analyses such as different quantitative tools. And so we were able to adapt the existing DSA mechanisms for our federated model of whole slide storage and tailor it for Alzheimer's disease and related dementia research and diagnostics. And so what exactly do I mean by harmonize? So as I briefly mentioned, since the DSA already had such a strong foundation, we were able to shift our focus on harmonization across the different sites. And so in this figure shown here, which is just demonstrating survey results from a National Alzheimer's Disease Research Center neuropathology core survey that was led by my colleague, Dr. Brittany Duggar. It just shows a heat map for the survey results from all of the centers for the brain regions sampled, shown here, in addition to the stains performed, shown here on the bottom. And so the purple indicates a higher number, and then the lighter blue and pale colors indicate lower numbers. So as you can see, there are some areas that the majority of the centers had the same sampling for brain region and the same stains available. So H&E, for example, for striatum was 100% of centers were sampling and performing that stain. So we really wanted to see what we had to work with, and we wanted to make sure that we were speaking the same language when we were referring to a brain region. So some places may sample a particular area and striatum versus basal ganglia. So in order to harmonize the language that we were using and the type of antibody that was being performed, this really helped us accelerate progress for aligning our metadata to be harmonized across sites. And so the BDSA enables the object storage and metadata management, taking this information such as brain region, stain performed, brain donor age, basic clinical diagnosis, and it also provides algorithms to process the whole slide data using Histomics TK. Plugins allow the system to add functionality to handle diverse data types, and there are DICOM plugin for large image plugin tools as well as annotation tools, and the job execution and management pipeline has been optimized. So again, this really has advanced to the harmonization stage for the metadata and allowed us to focus our time on completing data governance as well as algorithm development. So for data governance, I was lucky enough or unlucky enough to be one of the leads for completing this master collaborative data transfer and use agreement across multiple national as well as international sites. And we wanted to design this to enable sites to join the BDSA to be able to share their whole slide images and harmonize metadata in a federated manner, so that at each site, they would still maintain control of releasing the images on a project by project basis, and that the language would be as general as possible while still covering every aspect of compliance required by an institution for everyone to be able to participate and share their slides as efficiently and compliantly as possible. And so the BDSA specific goals include hosting and accessing the non-neoplastic brain autopsy whole slide images on a web-based open source platform to make it accessible for more sites, as many of the commercially available, even web-based softwares are quite expensive and require subscription type of arrangements where you have to pay for the service on a yearly basis. We also wanted to ensure that the BDSA was compliant with FAIR data principles, which include findable, accessible, interoperable, and reusable. And lastly, we wanted to develop workflows for sharing whole slide images, annotations, and metadata to enable and support training and deployment of algorithms on multi-institutional data sets. And so here I've just shown a couple of examples here. What's labeled as figure 3 is demonstrating the slide de-identification using our Python library resource, which enables one-click removal of the slide labels and metadata, which was developed by our BDSA team and led by Dr. Tom Pierce. And figure 5 is showing just a portion here of a whole slide image. And it's showing the proof of concept gray matter, white matter segmentation that is rendered on all of the whole slide images from federated sites. As shown from Kawai Dr. Chung's work, where the gray matter and white matter masks were generated on whole slide image data. So now to transition into combining the two U24s. So two separate U24 National Institute of Health grants were funded to support the development of digital pathology sharing and analyses in the field of neuropathology. And so one team is led by the individuals listed here, Drs. Gutman, Cooper, Duggar, and Pierce, and the other being led by myself and Dr. Pete Nelson and Cody Baumgardner. And it was requested that we actually unify these two projects to create an even larger expanded platform to support federated data management, whole slide image annotation, machine learning, and evaluation from central publicly accessible user interfaces. And it provides support for institutional and cloud-based resources, including a federated network. And the interactive and programmatic access to resources that we're working on will be made available to users both inside and outside our network, as it's funded explicitly by grants. And the merged digital pathology platform will be partially functional by year 4 of funding and fully functional by year 5. And so there was a lot of complementary expertise with the merger of the two U24s. And this is because there was different but complementary established expertise in each of the U24 groups. So I just have them kind of labeled as Emory for the one that's led by Dr. Gutman and Dr. Duggar, and UK for the one led by Drs. Baumgardner, Nelson, and I for clarity. And so the Emory group had made significant contributions to slide management, visualization, and annotation through their prior digital slide archive work focusing on cancer, whereas the UK group brought to the team significant contributions in the field to automated data collection, machine learning, and federated systems operation areas. And of course, across both groups, we have strong neuropathology expertise. And so we have been working to harmonize things using a new alpha beta testing plan. And the features and development for the BDSA that are primarily developed at Emory are then tested by the UK group, and the federated data resource management and training that's developed at UK is tested at Emory. So there's a back and forth flow for validation. And so the figures here are just showing, conveniently, we also had a long-standing history of collaboration between the two groups, as demonstrated in the table. And this is just kind of showing the overlap of expertise for metadata, neuropathology, and technology across the two different U24 groups for the collaboration. So this is just showing the BDSA Image Explorer, and I just want to explain a little bit about how this works. So it's web-based, and you're able to go on and select different filters depending on which type of whole slide image a researcher may be interested in. So you can search by Brock neurofibrillary tangle stage, CRAD neuritic plaque stage, age at onset, the primary neuropathologic diagnosis, and then you can search further by stain and brain region, and you get sort of this I guess it's like online shopping. So you have a little thumbnail of the whole slide image that would come up of interest once you enter your search criteria. However, you can't open the image. It's just sort of added to a shopping cart for your project, for example. And then when you select everything that you would like to include in a study, it then is connected to the site that provided the sample so that they get the information and are able to review and approve release of the whole slide images to the investigator through the federated platform. And this is just showing multiplex viz. So this is specifically for multiplexed immunofluorescence, and I'm just highlighting it here as this is kind of one of the newer areas that we're working on developing and optimizing, but it's exciting, and the BDSA will be able to support quantitative and spatial analyses on multiplexed immunofluorescent stained slides, in addition to the traditional bright field slides. So it's a very exciting tool that I'm looking forward to developing as we advance the project. So in conclusion, the BDSA is a work in progress, and there are challenges and opportunities involved with harmonizing the whole slide data and metadata across sites, but we've made a lot of progress. There's ongoing software development processes for the AD and ADRD whole slide images, and we are just finalizing the universal data use and material transfer agreements, which is exciting because we can get to working on some of the more fun stuff. And in addition to this, of course, performing the regular software testing. BDSA enables the development of secure, high-quality research tools while also maintaining control of whole slide image data at individual institutions through the federated design. And it is a strong foundation for standardizing shared scientific, diagnostic, and didactic applications across multiple sites moving forward. So this is the funding. Thank everybody on the U24 merger team, everybody in the lab and the brain bank in South Texas, and the individuals in my lab highlighted in red who have contributed most to the project. So thank you very much, and I'd be happy to take any questions.
Thanks, Dr. Flanagan. That was a great session. Next, we have the pleasure to welcome Dr. Gabor Kovacs. He's a neurologist and neuropathologist. He's a professor at the Department of Laboratory Medicine and Pathobiology at the University of Toronto, a consultant neuropathologist at the Laboratory Medicine program at the University Health Network, and a PI at the TANS Center for Research in Neurodegenerative Disease. He is also a senior scientist at the Kremlin Brain Institute, a faculty member of the Edmond J. Safra program in Parkinson's disease, and the co-director of the Rossi program for PSP research at the University Health Network. Dr. Kovac's major research interest is the neuropathology of neurodegenerative diseases. His team performs state-of-the-art morphology-based research using specialized microscopes and modern laboratory techniques to prove the neuropathological, biochemical, and genetic properties of different proteinopathies. In particular, they are interested in discovering the pathologic mechanism underlying nerve cell death and the reasons for the selective vulnerability of particular cell groups in tauopathies and alpha-synucleinopathies. Dr. Kovac, welcome to the ACE Global Research Summit.
Greetings, everybody who joined this webinar, and thank you, Marta, for the kind introduction. In this lecture today, I will focus on an overview of the molecular pathology of neurodegenerative diseases with a focus on Alzheimer's disease. So the objectives of this presentation is first to introduce the concepts of neurodegenerative diseases, then show you how we classify these conditions on a neuropathology basis, and then I will focus on the histopathology of Alzheimer's disease through the concepts that I introduce. So first, let me talk about the general concepts. I will focus on three aspects: the protein misfolding theory, the sequential distribution of proteins in the brain, and the concept of mixed pathologies. So when you see a patient with a clinical phenotype in the neurodegenerative clinic, either in the movement disorders or the cognitive neurology clinic, you have to think about vulnerability patterns in the brain of that individual. And these vulnerability patterns start with an anatomical vulnerability pattern, which functional systems are involved that cause the clinical symptoms. Then the cellular, which means which cell types are involved. Are these glial cells, neurons, which neurotransmitter cell types? And finally, based on the recent decades molecular discoveries, the proteins can have vulnerability patterns. So there is a misfolding of certain proteins, but there are various biochemical modifications which lead to which are associated with different clinical phenotypes. In the background of all of these, you have to think about the genetic background. Is there a disease-causing mutation or disease modifier mutation or variation? So the protein misfolding disease is a concept which identifies proteins as a center in the pathogenesis of neurodegenerative diseases, and that there is a mutation in the encoding gene or an unidentified pathogenic event that leads to the conformational change or misfolding of a certain protein. Now, this misfolding can lead to distinct folds of filaments, and these have been described recently using cryogenic electron microscopy. These distinct folds of filaments will propagate from cell to cell in a different manner, leading to system-specific sequential involvement of brain regions. These can be recapitulated in experimental models, and we call these strain-like properties because these different filaments will behave differently in the experimental models. This concept is also used for the so-called self-propagation, which we call seeding. And so-called seeding amplification assays have been developed that can detect these misfolded proteins. So in the clinical practice, you will be able to detect these in the near future. The cell-to-cell propagation is based on the discovery that the misfolding, when it happens in the cell, it leads to the formation of so-called oligomers, so a few molecules of bad molecules together, which can kill the cell or build up fibrils. If the cell is successful, maybe it can sequester these fibrils and build up intracellular inclusions. And this is exactly what a neuropathologist will detect under the microscope. Otherwise, the cell might fragment these fibrils and release them, so a next cell can take it up, and this is the concept of cell-to-cell propagation. Now, there is a very important further concept when we are talking about neuropathology of neurodegenerative diseases, and this is the so-called maturation of protein deposits. Maturation means that if we use immunohistochemistry and we use antibodies that detect disease-specific forms of bad proteins or pathological proteins, then we can see fine dots in the nerve cells. These are not yet ubiquitinated or not seen in silver stainings. These later, when they start to build fibrils and mature complex compact inclusions, that when we will see it with silver staining or ubiquity. This concept has been also described for the development of astrocytic or glial type of protein accumulation. So in summary, neurodegenerative diseases are characterized by misfolded proteins, and therefore we call them proteinopathies. We call the prion, when it is the prion protein, we call it PRP proteinopathy, which is the prion diseases. When amyloid beta, then A-beta proteinopathy, synucleinopathy, taupathy, and so forth. A very important conceptual discovery was the recognition of sequential involvement of brain regions in various neurodegenerative diseases. This means that if you look at thousands of brains, you will see a pattern. And the pattern means that somewhere the deposition starts in the brain and then sequentially involves further brain regions in a hierarchical manner. This has been described for all major neurodegenerative related proteins. And this is important because this opens the concept that there are preclinical cases. So that means that when the patient goes to the doctor, to the physician, with clinical symptoms, the brain deposition is already in a developed stage. So let's say alpha-synuclein is in the third stage from 6 when the patient goes to the neurologist, or tau is in the third stage from 6 when the patient goes to the neurologist with cognitive issues. Finally, the very important concept for understanding neurodegenerative diseases is the so-called mixed pathologies. Mixed pathologies means that different neurodegenerative diseases or other diseases, such as vascular diseases, are in the same brain. This is a study that we conducted nearly 10 years ago in a district of Vienna. It was a so-called Vienna aging study. And we found that irrespective of clinical symptoms, the individuals above 75 showed various combinations of neurodegenerative protein depositions with or without vascular changes. So the concept of mixed pathology has been recently crystallized, and this says that there are subgroups. One subgroup is that the brain contains different misfolded proteins, but there are no clinical symptoms. The other is that they do have clinical symptoms, and one pathology is very severe and the rest is not severe. Or there is a brain which has 2 or more severe pathologies. So you can understand that this will lead to very complex clinical symptoms. Indeed, and this is a review article which shows the large variety of different neurodegenerative misfolded proteins in the brains in the various conditions, showing that mixed pathology is very frequent and can lead to difficulties in the clinical practice to detect or evaluate the patients. Some studies also show that if there is low-level concomitant pathology in the brain, that increases the risk of transitioning from mild cognitive impairment to severe cognitive impairment by up to 20-fold. So this led to the concept of the threshold of leading to cognitive impairment. One is that if a patient has a pure Alzheimer's disease pathology, then the patient can reach this threshold. But the threshold can be reached also if the patient has low levels of pathologies, different pathologies, which together reach this threshold. Therefore, in the clinical practice, you will have to think about profiles, clinical, neuropsychological, neuroimaging, body fluid biomarker, genetic or other biomarker profiles that will lead to a better stratifications of patients for therapy trials, for example. In addition to increase the complexity, we have to think that the individuals are also aging. And during the aging, there are certain types of pathologies which are seen more increased frequency, and this will also contribute to the complexity of interpretation of symptoms if you are seeing more older patients in your clinic. So until now, I summarized the concepts of neurodegenerative diseases, and now I will focus on the classification, which has several levels: clinical, anatomy, predominant protein, and neuropathology. So at the neuropathology level, we are saying that there is a misfolded protein, there are maturation inside the cells of these proteins, and sequential distribution. So this is what we need to identify for one type of protein, but if there are combinations, we have to identify it for several different types. In the neuropathology practice, we try to identify which cell types and where in the cells do we see the misfolded protein aggregates. So this shows that the different major neurodegenerative disease proteins can be in very different cell types and show very different morphologies, leading to different clinical phenotypes. So as a neuropathologist, we do see a large spectrum of misfolded protein aggregates, how the differences in the morphologies and the anatomical distribution. And this is very difficult to translate to the clinical practice. Indeed, we try to classify the diseases not only based on the misfolded protein, but which cell types are affected and what type of clinical symptoms are these associated with, and are there any further aspects for classifications, such as biochemical modifications or genetic polymorphisms. Finally, recent cryogenic electron microscopy studies have identified that the different diseases are associated with different protofilament folds. So how the bad proteins misfold can be detected at an atomic level, and these are different in the various clinical phenotypes. After this introduction, now I will focus on the histopathology of Alzheimer's disease. So Alzheimer's disease is a very interesting disorder because it is two misfolded proteins. One is amyloid beta, and the other one is tau protein. Amyloid beta usually deposits extracellularly, so outside the cells, while tau is usually in the cells, so we say intracellularly. Let me emphasize that not all patients who have cognitive decline have Alzheimer's disease, although Alzheimer's disease is always associated with cognitive decline. Amyloid beta is a very complicated protein, but I will focus on two peptide types. One is the amyloid beta 40, and the other one is the amyloid beta 42. The 40 is released by secretase, or both of them are released by secretase, but the cleavage happens in a different part of the cell. On a neuropathology level, amyloid beta is either seen as amyloid deposits like plaques or in the vessel wall, which we call amyloid angiopathy. I will show you some examples. Here you see a little cord plaque in the left upper part, and then you see larger plaques with or without core. And on the right side upper, you see a vessel with amyloid deposits. So interestingly, the composition of these plaques can be very mixed with either the amyloid or both the amyloid beta 42 or amyloid beta 40, and these are found in different compartments of the plaque. And as you see, there can be different morphologies of the plaque. For example, this is called diffuse. The next one, what I show, is called fleecy type. The next one is vessel type, so cerebral amyloid angiopathy. And these are composed of predominantly different peptides. Interestingly, these different peptides also have different cryogenic electron microscopy structure, which also confirms that they have different folding of the protein. Now I would like to show you a few examples that the anatomical involvement leads to different morphologies of the amyloid plaques. So for example, we are now in the entorhinal cortex, and you see these diffuse type of deposits, but already starting to form a plaque-like morphology. Then if we go to a very close area, which is the pressubiculum, just very close to the entorhinal cortex, you see that the amyloid beta is kind of like a big lake covering this brain region. And then if we move very, very close to this, which is the CA4 subregion, or so-called cornu ammonis subregion, number 4, which is very close to the pressubiculum, you will see that the morphology of the plaques are very different. So they are much more plaque-like, and they have sometimes a very big and identifiable core composed of amyloid. Or, for example, if we go to the basal ganglia, the putamen can show this diffuse type of deposits. This is the striatum, while the globus pallidus, which is just adjacent to the putamen, will show these little core-like amyloid deposits. So with this, I wanted to show you that even in the same brain, the morphology of the plaques can be very different depending on which brain region you look at. So if you are using, for example, positron emission tomography, these morphologies will be picked up differently depending on what ligand you are using. Now let me talk about the sequential distribution of amyloid. This has been first mentioned by Brack and Brack many decades ago, and they identified 3 major stages. However, in a 2002 study, this was more described in more precision, and these are now called phases of amyloid beta deposition, and because of the first author, it is called TAL phases. So in the first phase, the amyloid deposits in the neocortical areas, especially the frontal, temporal, and occipital neocortex. In the phase 2, the entorhinal region and the CA1 subregion of the hippocampus and the insular cortex is affected. In the third phase, the subcortical areas like basal ganglia is involved. And in the fourth phase, the brain stems such as the midbrain, the substantia nigra is affected. And finally, in the fifth phase, the cerebellar cortex also shows plaques. The other protein in Alzheimer's disease is tau. Tau is present in the normal brain, and we have 6 isoforms. 3 of these 6 isoforms have, in a certain part of the tau protein, 3 repeats, and 3 of them have 4 repeats. And interestingly, in tau-related diseases, either the 4 repeat isoforms or the 3 repeat isoforms accumulate in the inclusions. But in Alzheimer's disease, interestingly, both. So both the 3 repeat and the 4 repeat isoforms accumulate. Another important aspect for tau diseases is the phosphorylation. So we see a much more phosphorylation in tau diseases. For example, we have a lot of phosphorylable epitopes in the normal brain, and then these become phosphorylated when we have a disease. Alzheimer's disease, in the current modern classification of tau diseases, is classified as extracellular filamentous deposit-related tauopathy, which means that in Alzheimer's disease, there is an obligatory presence of amyloid when you see tau. Brack and Brack initiated the staging of the tau pathology in 1991, and this was updated using modern immunohistochemistry methods. The original one was done with silver staining. The original identified 6 stages, starting in the transentorhinal cortex, so in the hippocampus area, expanding to the limbic areas, and then to the isocortical in the last stages. Using immunohistochemistry, a much easier method was developed, and this focuses on the so-called neuropil threats detected by 88 immunohistochemistry and a semi-quantitative scoring. We focus on the following regions. the hippocampus block, transentorhinal, entorhinal, occipitotemporal cortex. So this you can get in the hippocampus block. Then the middle and the superior temporal cortex, which is a second block to be stained. Then the occipital cortex, which is the third block to be stained. So with these areas, we are able to stage the Braak stages. However, recently, Braak and colleagues identified that the brainstem might be before the entorhinal cortex, and this is now called 1A or 1BC stages. Finally, for the full diagnostic procedure of Alzheimer's disease, we still have to evaluate semi-quantitatively the frequency of so-called neuritic plaques in the cortex. And then this leads, in the neuropathology practice, the final neuropathology diagnosis, which is called ABC score. And that means that by the combination of the amyloid tau phases, the Braak stages, and the SERAD neuritic plaque score, we are able to identify the level of AD neuropathologic change from low to high, and there is an intermediate phase. And this is what can be better discussed for clinical pathological correlations. In the last 2 minutes, I would like to return back to the concept of mixed pathologies, but now focus on Alzheimer's disease. Here I list the 3 most frequent mixed pathologies in Alzheimer's disease. This is vascular disease, Lewy body diseases, or limbic predominant age-related TDP-43 encephalopathy. I will touch each of these topics. The first is the vascular diseases. This is a very complicated topic, and actually neuropathologists do not necessarily agree how to evaluate this. Therefore, there is a discrepancy in the literature. Also, there is a discrepancy because many neuroimaging studies, MRI studies, describe a lot of vascular lesions in cognitively impaired individuals and call this vascular dementia. However, MRI studies do not evaluate, for example, TDP-43 or even Alzheimer's disease pathology. Therefore, they cannot really exclude whether which pathology really contributes to the clinical symptom. In addition to infarcts, we have to focus on vessel pathologies, and this can be the arteriolosclerotic small vessel disease, but also diseases affecting the venous system. The next topic or frequent copathology is Lewy body diseases, and these are well known, and there will be also webinars introducing this topic. But this can be distributed only in the amygdala, limbic system, or they can be in the brainstem and also in the neocortex. Finally, limbic-associated temporal limbic predominant age-related TDP-43 encephalopathy, which was already described earlier, and 6 stages have been recognized for the distribution of TDP-43 in Alzheimer's disease. However, in 2019, this was conceptualized as late, and 3 major stages have been recognized. Importantly, late can be associated with hippocampal sclerosis, which is a so-called hippocampal sclerosis of the aging, and it is not the same as that seen in hypoxic damage of the brain or after epilepsy-related conditions. We have to emphasize that TDP-43 can be seen in all of the hippocampal sclerosis cases, but can be seen in cases in late without hippocampal sclerosis. So the conclusions of my presentation is that neurodegenerative diseases are characterized by deposition of misfolded proteins in the brain. Neuropathology reveals different morphologies and cellular involvement and suggests subtypes of conditions. Recent studies reveal the molecular background for this variability. And very importantly, mixed pathologies are frequent in the aging and neurodegenerating brain. Thank you for your attention.
Thanks, Dr. Kovacs, for a very interesting session. And last, we have the pleasure to welcome our third speaker of the afternoon, Dr. Michael Henneke. Dr. Henneke is the director of the Luxembourg Center for Systemic biomedicine at the University of Luxembourg and PI of the Neuroinflammation Group since 2022. He is a board-certified neurologist with a long-standing interest in immunology and neuroscience. He has broad experience in the study of neurodegenerative diseases at experimental, preclinical, and clinical levels. The main focus of his research is related to dementia and AD, but he also works on ALS, PD, MS, and septic encephalopathy. His clinical practice, Dr. Hennecke, holds special expertise in neurodegenerative and autoimmune CNS disorders. Dr. Hennecke, over to you.
Well, thank you, Marta, for this kind introduction, and hello, everybody, to this Friday afternoon session. I want to discuss with you in the following presentation how innate immune activation shapes the pathology and shapes the phenotype of Alzheimer's disease. And we start with looking at images that had been painted by Alois Alzheimer and Franz Nissl more than 100 years ago. And as you can see here, they already depicted the massive astroglial and microglial reaction to the pathology. They called microglia cells amoeboid glial cells, shorted by AGLZ, as you can see in these pictures, and place these immune cells at various positions around the amyloid deposit, close to neurons, and at sites of brain vessels. They also depicted, as you can see in the middle picture, the death of these cells and the suffering in response to immune activation. Now, this is showing you a cortical section stained with Congo red and MSG class 2 for microglia cells. And actually, Congo red has been long before being used as a stain for human brain, licensed as a bacterial stain, raising the question, what's the commonality between a human brain and a bacterial surface? And it took about 100 years longer until Matthew Chapman described that various bacteria common to us, including Escherichia coli, Staph aureus, Salmonella typhimurium, carry beta sheet structured amyloids on their surface, looking pretty similar to what's being deposited in the brain. So that means to a myeloid cell, including microglia cells, beta sheet structured amyloid means a pathogen, and nature has, through evolution, equipped these cells with various pattern recognition receptors, including the toll-like receptor system, CD47, CD36, and other surface receptors that can sense the presence of beta sheet structured amyloids and react to them. There are mainly two reactions. One is activating the innate immune response, and one is phagocyting the opponent and clearing the tissue. So if microglia cells gets activated, that's very early and in response to beta amyloid deposition, which we believe now takes place decades prior to the onset of clinical symptoms. And because of that, we have to acknowledge that after the deposition of beta amyloid and activation of the innate immune system, every mechanism, any pathogenic event after will be happening in the presence of inflammation. Now, if a microglia cell gets activated, it undergoes a massive change of its phenotype. It withdraws its processes, which usually are being used to scan the immediate environment. And you see that all these processes are withdrawn. They start to release a lot of inflammatory mediators, including complement factors, cytokines, chemokines, radical oxygen species, and nitric oxide. At the same time, they stop doing a lot of important things for our brain. They reduce synaptic scaling. They reduce the production of trophic factors, including BDNF and others, and they get less active in debris clearance. Now, neuroinflammation, or the presence of immune mediators in the brain, has a functional consequence. As shown here, you see that a single cytokine such as interleukin-1 beta can suppress long-term potentiation, which is a very important factor for information storage and consolidation. And And this is not the only cytokine that has this detrimental consequence. There are others, including TNF alpha, IL-6, interleukin-1 alpha, C1q, and C3. So many of these immune factors can actually impact on neuronal functioning. Now, beyond that, there is a structural consequence. This is work by Guy Brown's lab, who showed that microglia cells can take up neurons when they do not present so-called do not eat me signals. So this is a mechanism which involves the MFG-E8 receptor and other mechanisms. And shown here is that there is an experiment where microglia cells have been stimulated by amyloid beta 1 to 42, about a 30% loss of neurons in the cell culture dish that cannot be attributed to necrotic or apoptotic death, but that's due to the uptake and of these neurons by phagoptosis. Now, there is another mechanism that has been identified by Suyeon Hong and published in Science a couple of years back. And what Suyeon did was she labeled synaptic structures with Homer GFP and found those back into IBA1 positive cells. So in APP-PS1 animals, there is an excess of uptake by microglia cells of these synapses, and this is happening in a complement receptor 3 dependent manner because the knockout doesn't show the same phenotype. So this all happens on the background of our lifestyle and various behaviors. We know that immune activation is driven and massively influenced by peripheral factors such as midlife obesity in men, which is a risk factor for developing Alzheimer's disease, poor oral health, periodontitis, aging itself by producing a senescence-associated secretory profile, which is the low-level release of various cytokines. Brain trauma has a direct impact on inflammation in the brain. Infections or systemic inflammation is a driver of immune processes in the brain as well. And reduced physical activity is a factor which elevates cytokine levels in the blood, which are likely to impact on brain immunity as well. All this comes on our genetic background, and over the past decade or so, we have identified a number of risk variants, of which many directly impact on innate immune signaling pathways, including TREM2, APCA7, but many others as well. Now, it doesn't yet tell you in which cells these risk variants play an important role for disease pathogenesis, and this has been alluded to by Kozo and colleagues in this very elegant Nature Genetics paper from August 22. And what this group from Columbia University did is they used an attack sequencing approach, taking microglia cells or material from microglia cells from 150 brains, and then did a transcription footprinting approach, run GWAS information against it and RNA sequencing data, and basically identified in which cell GWAS variants would have an important role. So you see on the right side, new and positive cells for neurons and microglia cells. And if you just focus on the last 3 neurodegenerative diseases, which are listed here, in Parkinson's disease, it looks like there is no significant role for any risk variants in neurons. While in ALS, that's highly significant, meaning that risk variants really play a role for neuronal changes in this disease. In Alzheimer's disease, there's an even negative signal for any GWAS role in neurons. While moving to microglia cells, you see there's a slightly but significant role for GWAS identified risk variants in Parkinson's. In ALS, there's the trend towards it, but the variability doesn't allow for a significant result, while in Alzheimer's disease, there's a highly significant result, meaning that the jivas that had been identified so far may play an important role, but in microglia cells or by influencing microglia cell biology. Now, this shows you an interesting comparison between pre-symptomatic stages, the MCI stage and the AD stage, with respect to PET imaging for amyloid, tau, and inflammation. And what you see is that as soon as you get amyloid beta deposition in the brain, you have concomitant inflammation already in the pre-symptomatic stage of the disease. And in the first part of my presentation, I now want to show you how these factors are influencing each other. Now, it's very important when we study these things to really restrict our views on what is known about human immune function in the brain, because a high number of pathways are not being conserved between mouse and man. And you know that we use a lot of mouse modeling for analyzing these factors. So this is work by Eric Bodek and colleagues who identified a core signature of human microglia cells, comparing 4 different data sets. And in the core of the core signature, you find the inflammasomes and their products. And we were interested specifically in the NALP3 inflammasome, which is immune signaling pathway upstream of many others, requiring ligation of a cytokine receptor or toll-like receptor that produces the proforms of cytokines and chemokines, as well as the auto-inhibited NLRP3, followed by a second step of activation, which is licensing the buildup of the active NALP3 inflammasome in response to mitochondrial damage or lysosomal damage, potassium shift, or re-exposure to amyloid beta aggregates, or, for example, silicium crystals. Now, the maturing step involves the formation of an ASC-SPEC, which has outward-directed active caspase activity that cleaves pro-Al1 beta, pro-Al18, and gasdermin D in a manner that would allow the gasdermin D cleavage product to form a pore complex in the outer membrane, allowing for the coordinated release of these cytokines. And we have identified this system in human AD patient brains, shown here an active ASC-SPEC in a frontal cortex microglia cell of an AD patient. And you can take the cleaved caspase-1 band as an activity signal. Comparing this in human brain in frontal cortical sections between age-matched and non-demented controls to 80 cases, we found that there would be a massive increase of NALP3 inflammasome activation in these brains. And luckily, this is also being mimicked in the mouse brain. If we cross APP, PS1 mice into NALP knockout mice, we completely suppress the cleaved caspase-1 band. Looking at the function of these cells by knocking down NALP3 and interleukin-1 beta, we would massively improve phagocytic clearance of amyloid beta. We would protect synaptic density and would reduce A beta and spine loss. At the same time, we would protect the animals from LTP suppression and spatial navigation deficits in a memory test. Now, the question is now, how does the system impact on the structure beyond spine density? And so we were interested what would happen with these cells if they would be chronically exposed to amyloid beta. And one phenotype is that microglia cells and immune cells die from an hyperinflammatory death called pyroptosis. And what you see here in the next movie is one of these cells releasing an ASC-SPEC at the moment of death, which immediately binds red-labeled amyloid beta. So the beta amyloid forms a halo surrounding the ASC-SPEC. And we control this also by electron microscopy, showing you on the left side, you see amyloid beta 1 to 42 alone, the ASC-SPEC alone in the middle, and on the right side, I hope you can appreciate that the ASC-SPEC acts like a magnet to the amyloid beta peptides, suggesting that once a homeostatic microglia cell would undergo activation and during chronic exposure and chronic inflammation die from a pyroptotic death, the released ASC-SPEC would be an ideal matrix for amyloid oligomerization and eventually the deposition of beta amyloid. And we tested this in this very easy cross-seeding assay, which shows you the conversion of a monomer to an aggregate. In green, you see the aggregation of amyloid beta over time in the absence of any other factor. And then we add in ASC-SPECs in increasing concentrations, which induce this massive shift to the left, which is a classical sign of cross-seeding. We also tested this in a blot system, showing you that in the absence of any ASC, we hardly have oligomer or fibril formation, which is increasing in response to adding in more and more ASC into the same experiment. You see the occurrence of A-beta oligomers and fibrils. Now, this is the same for amyloid beta 1 to 40 as with amyloid beta 1 to 42 shown in the second plot here. Now, testing this further, and this is a co-sedimentation assay that has been performed in collaboration with Jochen Walter and Satish Kumar in Bonn. And what they did is they looked at amyloid beta monomers 1 to 40 and 1 to 42 amino acid length, either alone or together with ASC. And ASC alone, you see that at time point 0, everything is in the supernatant. At 6 hours later, everything has moved in the pellet, indicating that the ASC-SPEC would have drawn all the amyloid beta peptides into a kind of aggregate or conglomerate. And this prompted experiments where we tested mouse brain, and we did an immunoprecipitation experiment for ASC and blotted for amyloid beta, and we found that amyloid beta would bound to the ASC-SPEC here. More importantly, if we look at the histology of these amyloid beta deposits in these animals, we would see that there is an ASC core and the formation of a 6E10 positive amyloid halo surrounding it, very similar to what we had observed in cell culture. Now, proving that further, we employed a mouse model that has been introduced by Matthias Jucker and Larry Walker, which uses the injection of an APP brain lysate versus a wild-type brain lysate into opposing hippocampy and studying the spread of pathology after a certain time. This model has a problem because synthetic amyloid beta is not seeding, suggesting to us that either the amyloid beta is different or there is one or more endogenous cofactor. Now, in this model, we found that if we would inject APP-PS1 derived mouse brain lysate versus wild-type brain lysate, we would completely confirm the previous claim by Matthias Jucker in his laboratory, showing that there is a twofold increase in A-beta deposition, and the hippocampus that received the APP-PS1 brain lysate, non-injected brain in comparison proved that wild-type brain lysate injection would not have any effect on the system itself. Looking to the blots, you see that APP processing is not affected at all. Total amyloid beta is massively enhanced, as is the A-beta oligomer amount. And now we repeated the entire experiment on an ASC knockout background, showing that there would be no such increase in A-beta deposition, and most importantly, no increase in total amyloid beta or oligomer formation, suggesting that oligomer formation and deposition in response to APP-PS1 brain lysate injection depends on the presence of this inflammatory molecule, and in particular, the ASC-SPEC. Now, how does that look in humans? We tested human 80-patient brain compared to age-matched controls, and here you see that we see the same binding of A-beta to ASC. And if you compare this across controls, MCI patients and AD cases, we see that already in the MCI stage, we see in the core compartment of the plaque, amyloid beta and ASC co-localized, where the only difference of the AD patients is that we have more amyloid beta in the fluffy fiber compartment of the plaque. And looking at the histology, again, as in the mouse model, we found the ASC spec positive core of the plaque in red and the 6E10 halo surrounding it. Now, this is not all, and there is more action of inflammation on AD pathology. And here I show you that we now are interested in how we get from the pre-symptomatic stage presenting with amyloid and inflammation to the tau pathology. And this was tested in a mouse model where we adopted an experiment that had been introduced by Jürgen Götz and published in Science already in 2001, using the same APP-PS1 brain lysate, but injecting it into a hippocampus of a tau mutant mouse, the tau22 mice, in comparison to wild-type mouse brain tissue. And doing so, we found that we would increase the occurrence of AT8 positive tau staining in the hippocampus of tau22 mice, but on the ASC knockout or NALP3 knockout, that would be completely absent. And here you see the quantification. So the red bars compare the tau22 animals. You see the clear and statistically significant increase of pathology, which is absent in ASC knockout or NALP knockout backgrounds. And to make a very long story short, we identified a pathway where pattern recognition ligation and A-beta exposure to microglia cells would be followed by interleukin-1 beta and IL-18 release, and in an interleukin-1 receptor, MyD88, MEC1/2 dependent pathway. This would result in CAM kinase 2 alpha phosphorylation, which subsequently would phosphorylate tau and leading to the formation of paired helical filaments and neurofibrillary tangles in these neurons. So in summary, I hope I could show you that neuroinflammation is an important component of Alzheimer's disease pathogenesis and pathology. Neuroinflammation impacts on the function and structure of the brain. Lifestyle behaviors and genetic background are influencing neuroinflammation. NALP3 activation drives A-beta deposition in vitro and in vivo through ASC-spec seeding, and NALP3-derived interleukin-1 beta drives tau phosphorylation in neurons and contributes to tau pathology and ultimately neuronal death. And with this, I'm thanking the people of my laboratory, which have been involved, as well as our great collaborators and the funding agencies that support this work. And thank you for your attention.
Thank you for a great talk, Dr. Seneca. So now we have completed the 3 talks of this afternoon summits. All of them have been very interesting, but also very different, offering us a great overview of the neuropathology of Alzheimer's disease from different points of view, covering from digital pathology to a great description of AD histopathology, and lastly, the role of immune activation in the brain. I would like to encourage our audience to start sending questions to our speakers in the chat. I don't see any questions yet, so I'm going to start myself. So if we can see all our speakers. Okay, so I'm going to start with an open questions, maybe for Maggie and Gabor. I'd like to know what are the learnings from your wide experience in necropsy studies from AD patients that could help us clinicians like myself in diagnosing and treating better our patients and developing better biomarkers. So we know that neuropathology is the gold standard for the diagnosis of neurodegenerative disorders, but you can see many things that we clinicians do not when our patients are still alive. So you have reviewed how complex the changes found in the brains of AD are, but most of the times our patient just receives a diagnosis of AD. Gabor, for instance, you mentioned about developing different profiles for stratification of patients. What would you like us to do better in clinical?
Thank you. I don't know, Maggie, if I should start or you want to start.
Please go ahead. Yeah.
Okay, thank you. So thank you for this question. In my opinion, the situation is not that bad generally. So I see very optimistic changes in the science in this respect. So I think that we have two approaches to consider. One is to identify the pathogenic proteins in the brain, and second, to identify what is the dynamics of the neuronal degeneration and how the neuroinflammatory components look like. So to identify the pathological proteins, already there are very promising markers and already used from CSF for amyloid beta and tau, and it seems that this is now kind of translated to blood. And this is good because the CSF might not be easily approached for a lot of patients, maybe in big centers, yes, but not for a lot of patients. The other things are the copathology is the most frequent, is synuclein. And it seems that for synuclein, the so-called RT-QuIC, which is a seeding amplification assay, is becoming very promising in the clinical practice in Parkinson's disease patients. And this is now started to be used also for kind of cognitive decline patients to exclude the presence of Lewy body type of pathology. Currently, this is done from the cerebrospinal fluid, but there are very promising from the very tiny skin biopsies also. And there are big efforts to try to do this from blood even. And there are reports which succeed. The question is that how soon this can be reproduced by other laboratories and how soon can this be widespread in the clinical practice. The most difficult currently may be this TDP-43, which is a very frequent copathology, especially in Alzheimer's disease. There are studies which suggest that maybe even there the seeding assay might work. The question is from what type of body fluid or eventually skin biopsy. However, we should not forget that still the neuroimaging is a very important component, and detection of atrophy patterns in the medial temporal lobe can be very suggestive of one or these conditions. Eventually, the hippocampal sclerosis, as I mentioned, is very, very associated with TDP-43. So if there is an asymmetric change, that could be already very supportive. I still believe that the detailed neuropsychology profiles also reflect these differences. So I think that these very precise clinical examinations already can be suggestive of a concomitant disorder. And finally, to identify how fast is the disease going, that's what I meant by markers for neuronal degeneration or neuroinflammation or other pathogenic events. I think that there are studies which try to explore this. It's not yet translatable to the clinical practice. First, these have to be standardized among the centers which report these blood or CSF-based markers. Currently, the neurofilament light chain is what is used for, which can be used from blood actually for neuronal degeneration. But we do need other markers which reflect the neuroinflammatory process. So thank you.
Thank you. Gabor. Maggie, do you want to add anything from your point of view?
That was very comprehensive. I don't really have too much to add other than, I guess, just commenting that there are also some emerging retinal biomarkers that are also being developed. But again, they require validation and are still works in progress. But yeah, very comprehensive. So not anything else beyond that.
Cool. So we have a question for Michael from the audience. So from Haritha Katagada. So are there any specific markers to differentiate between acute and chronic inflammation?
So in general, assessing inflammation is very difficult because most of the acute markers are very short-lived, and we're still seeking markers for who are practically giving a fingerprint of what had happened as an inflammatory insult, especially in these diseases who have year, if not decade-long trajectories. And we clearly see that some of the markers, as the TAM receptors, for example, come up in very early stages, even in what we call healthy controls with already pathological signs or the SCD state, and then burn out in MCI in 80 cases. So while other markers who are more prominently describing the chronic state of inflammation are arising in the 80 patient brains. So there are no established markers. The ATN criteria, as all of you know, have been expanded to inflammation as one further component. But I believe, to be honest, that YKL-40 or GFAP are really bad markers for inflammation and not reflecting what a microglia cell actually does. It's markers for astroglial reactivity, which are very unspecific for Alzheimer's disease. I think it will end up with having a plethora of markers which now are being identified by Olink panels and more sophisticated proteomics. And I believe that by comparing several data sets which have been accumulated very recently or are being accumulated right now, we will probably in 1 or 2 years have a clearer view on which stage of Alzheimer's disease is characterized by which type of inflammatory profile.
Okay, following on that, so Michael, you talk about that all the events happening after a beta deposition in AD are going to occur in the presence of inflammation, so highlighting how important this is. And you were talking about that you are not a big fan of the actual biomarkers that we have for that. What is the ideal biomarker that you would like to have for active immune activation in the brain?
Yeah.
And yeah, you can answer that if you want.
Yeah, it's already a complex answer, because either I would have to have something that freezes these short-lived acute markers like the interleukins, the complement factors, the chemokines, which are by nature very short-lived, or I would like to have, as I said, a kind of fingerprint, a post-translational modification that is stable and it's not being changed. So nitration of tyrosine residues in response to immune-stimulated nitric oxide generation is one possibility, but there are others. And so one would have to have a marker of which we know that is unchanged once it has been kind of imprinted. And on the other side, this is kind of the contrary, because inflammation is usually shut down very, very quickly in order not to harm the host, and it's a defensive reaction. But we learn that over time and chronicity, as in many other chronic inflammatory disease stages in other organs or in the skeleton, or you name it, it has a detrimental action on the host tissue.
So Michael, apart from improving our lifestyle in general that we all should do you have any anything you would recommend we do in different stages of our life to reduce this inflammation?
Well, I think the Lancet Commission has put out a great manuscript last year, Livingston and colleagues, mid-24, with 14 factors, which we all can address already within some more easily, others a bit more difficult. But, by working out, by adhering to a Mediterranean diet, by not smoking or drinking too much alcohol, these are the easy things we can, take over in our daily lives. And that will, if we would address, I think, all the factors listed there, we would probably have the number of patients in 2050, and that would be a great relief for our healthcare systems globally, I guess.
Sure. So, okay, while we wait for more questions, I'd like to bring on a topic about brain resilience in Alzheimer's disease. So individuals who die at an elderly age who are cognitively intact but have maybe plaques and tangles in their necropsy. Maggie, for instance, do you have much experience in this topic? Do you think that this concept is really true, of these individuals have lived longer, they would have developed dementia? because the new Alzheimer's Association criteria labels everybody with a beta biomarker as having AD, and they believe if they died earlier, and if they have lived longer, they maybe would have developed AD. What is your point of view from the neuropathology world?
Yeah, great question and great topic. So, in the experience working with the NUN study in Honolulu, Asia, aging study cohorts, we found that so-called resilience, so those who have the high plaques and tangles but were cognitively intact, were actually found to have less coexisting pathologies. So, less coexisting Lewy body disease, less TDP, and less vascular lesions. However, I do think that there is a small subset of these so-called resilient individuals that there may be something else going on, more downstream perhaps related to neurotransmission. So I know that there is some emerging research in that area that was presented at the ADPD conference. So I think that that's an exciting new area, but I think for, at least in my experience, the majority seems to be could be largely explained due to the less frequent coexisting pathology lesions other than plaques and tangles.
Thank you. Following up that, Michael, do you think about if the different levels of inflammation in the brain on these resilient patients could have a role in this factor?
Yeah, I think I cannot prove that, but if I remember correctly, there have been very old papers in, I think, around 84, 86, also coming from the NUN study, who showed that exactly those persons who had developed dementia also had glial reactivity and microglia activation surrounding the plaque deposits. At that time, I think that was work by Joe Rogers and colleagues and others. And the microglia markers were really, they used what was available at that time. But sometimes I think that I would love to substantiate that work even further with modern markers and modern technologies, and eventually doing in situ hybridization and transcriptomics to shed more light on that. But that was actually one of the suggestions, that it would be the inflammatory reaction which would, at least on a pathological level, distinguish the patient population which would develop dementia from those who were not. And I see Margaret shaking, nodding and agreeing to that, but maybe she has more comments. I think that's clearly her expertise in her area.
Yeah, I think those are all really great points and things to build on from older research with less sophisticated techniques. So I do think it's a great opportunity in the current time that we're in, that we, I think, really have a new chance to explore these things in more detail and really find clarity. And I think it's going to be quite interesting to look at the neuroinflammatory profiles in the settings of, plaques and tangles by themselves, and then also, of course, see if there are resilient individuals who seem to perhaps have resilience even in the setting of multiple lesion types, and that there could very well be many different inflammatory differences in the different, I guess, combinations of disease, which will be fascinating. So that's definitely something we're working on expanding, but don't yet have answers. Yeah.
Cool. So we have another question from our own Dr. Agustin Ruiz, also in San Antonio now. So his question is, do you think brain banks are instrumental for dissecting genetic risk factors, or are pathological phenotypes too evolved in postmortem subjects to research the etiological aspects of neurodegenerative diseases? Who wants to take this?
I mean, I can start, Maggie, if you think. Yeah. So thank you for this question. Of course, one of my answers is that, of course, it's very much needed. Brain banks and brain donation. I do think that when we look at the brains and start to use digital pathology and kind of new methods for statistics, clustering, analysis, and so on, then we see that even if we call the diseases the same name, like Alzheimer's disease or progressive supranuclear palsy or whatever, the patterns in the brain can be very different between patients. And that, for me and for others, already suggests that the etiologies can be very different, leading to the same pathology. And I think that this is a very important component. And from a well sampled brain, you can do a lot of things. So high quality transcriptomics, high quality genomic and other omics studies. And if we pool enough cases and we have good clinical data and using the modern technologies, especially bioinformatics, then I'm sure that this will reveal already very different patterns. As for an example, when we looked at the seeding or molecular behavior of synuclein or tau in Parkinson's disease or progressive supranuclear palsy, just based on the molecular behavior, we could differentiate patients. And then when we sent the samples for proteomics, the completely different patterns came up for these patients, which means that the pathogenesis is probably very different, leading to the common pathologies. It is very important in brain banks, and that's a little bit a critical aspect, is how we do the sampling and how we do the neuropathology. So it should be very standardized, and not all brain banks are standardized in this sense around the world. And I also am a little bit critical also that one hemisphere is examined, the other is not, because I do see a lot of asymmetry in these pathologies and also in the clinical practice. So I think that these are the kind of the cautionary notes, but generally, of course, it's a very much needed thing. Thank you.
Thank you, Gabor. We have another question from Hatis Rekayoglu. I'm sure I said that wrongly. So she says, a small proportion of the Down syndrome patients also do not develop Alzheimer's disease. May I learn whether this is also related to the coexisting pathologies, or there is another explanation for that?
I can try to share what I know in this area with the caveat that I'm not a major Down syndrome expert, but if any of the others I will offer first before going for it.
Please, Maggie, start.
Okay. So from what I know of the literature in patients with Down syndrome, neuropathologically, it appears that they do develop similar frequencies of coexisting pathologies, particularly TDP43, which is quite interesting. So I'm not entirely sure that with those findings that have been published, that would be an explanation for the subset that do not develop Alzheimer's disease. I would suspect that it would be due to something else. Yeah.
Any other ideas on that?
Yeah.
I mean, just my own experience on adult Down syndrome cases, that I also see these copathology patients. And about 10% of the brains that I examined with adult Down syndrome had very low amount of Alzheimer pathology and no copathology, but the age was a little bit lower, so like 50 instead of 60. So maybe if we say that there are Down syndrome patients who don't develop Alzheimer disease, maybe the age category, it might be important, or any other genetic factor that might influence, or even the sex of the patient that we have not yet probably evaluated systematically for Down syndrome.
Okay, so while we wait for other questions, Howard, you mentioned about the different types of A-beta plaques that can be found in several regions on the same brain with Alzheimer's disease, which is fascinating to me. Can you elaborate on how this difference can influence the clinical presentation of the disease? And do you think they can influence the response to the novel anti-amyloid therapies?
I mean, thank you. I think that these amyloid deposits, and this is true for other protein deposits, is the local environment defines how the cytopathologies or the extracellular protein deposits develop. And very recent studies show that if you microdissect these plaques, for example, then the molecular composition can be very different, which means that, again, supporting the local environment. I think that the basic core features are probably the same, but then the further modifications can be different. I don't know how this would affect the therapy because it depends on what exactly the therapy is targeting on beta amyloid or whatever protein. Where I think it might be more interesting is, for example, in PET imaging, because some of these deposits might not be picked up by the ligands that you use, because the ligands might stick to the end matured amyloid structure, but there are fibrils which are not yet reaching that type of beta-sheeted amyloid structure that might be picked up with a certain ligand. So here you might have differences in the brain regional distribution of PET studies.
Okay. Now for Maggie. So you talk about digital pathology and your big project that you are co-leading that is now trying to be used in order to harmonize the data, use artificial intelligence for the analysis, and allow federated sharing of these slides. So how do you think this digital pathology will change the future of neuropathology and help advance in the discovery of novel entities and biomarkers?
Yeah, it's a great question. So how will digital pathology propel the field forward in the research areas of neurodegeneration and biomarkers? So I think that first, it allows easy sharing of cases. So I think that by increasing the number of samples that are available to use as training and validation samples will only improve algorithm development and make the harmonization by neuropathologic classification more standardized and more objective across sites. So as Gabor mentioned, sometimes sampling even can be very complicated and differ by different institutions. So by having this project and at least starting at a point where we can all check, okay, what do we all sample the same? What do we all stain the same? And then sort of from there expanding out to see if there are slight differences. Can we compare exactly where the sampling was performed? For hippocampus, for example, more anterior versus more mid or posterior, to really kind of test what can be, merged and considered the same region versus what should be separated out and called something else. I also think that having a digital pathology federated open source platform will really be helpful for advancing research using AI specifically. So I think that all of us have bias, and by just having more objective tools to quantify, how much of a protein, in addition to being able to assess XY coordinates and different cell populations, that this will enable us to identify and delineate disease mechanisms related to proteinopathies and neuroinflammation, and better understand the cell-to-cell interactions and impact on neuronal function and synaptic connectivity in a more objective manner. So I think that these are all really big things that digital pathology is going to be able to support and build a foundation for to rapidly advance research. And in addition to that, I think it provides a resource that was previously considered to be non-renewable is now never going to be non-renewable, at least for these types of studies, so that all researchers can analyze the same whole slide images in different ways, and that that in and of itself will enable us to find more research findings than we would have otherwise if, for example, a block were exhausted or something like that for, a rare case, we'll say.
Okay. So, Gabor, you mentioned that there are several pathologies that are found in aging brains, not necessarily with cognitive decline yet. So how are these different from those pathologies found in brains from people with cognitive decline and dementia, and why?
I mean, this is a very difficult topic, and I think that some of these are very neglected things, like even in the neuropathology practice. Let me give an example. In 1960s, there was a Professor Hirano in Japan described the Hirano bodies. Nobody cares about that in the neuropathology. And then one of the Japanese colleagues who were visiting my lab, then he sat there for 1 year and counted all the Hirano bodies in all the brains we had. And then he found that the Hirano bodies are very different in progressive supranuclear palsy than in Alzheimer's disease. So then we started to think that maybe, you know, these unknown, or not unknown, unrespected or underappreciated aging pathologies do have a message. So that was one thought. So yes, so these aging-related pathologies probably reflect a kind of all the effects that we get during our life, and our brain responds to that, be it a glial response or an inflammatory response or whatever things happen with us in our brain, and then our brain generates these kind of pathologies, and these mostly affect astrocytes or the corpora amylascia, which is again a very underappreciated pathology, is the vastosomes, and it now gets much more attention with the focus on G lymphatics and other systems. So these are the age-related pathological alterations, and a very interesting thing is that as older individuals get, they tend to generate more tau pathology and not beta amyloid. So if somebody lives 100, 120, will have tau pathology mostly in astrocytes or glial cells and not necessarily amyloid plaques. So these are really the successful agers who will not really develop Alzheimer's disease. And also, if you look at the San Diego Zoo, the very old baboons will also generate glial tau pathology and not amyloid plaques. So it means that how we age, the kind of the, let's say, normal or whatever aging type of pathology response can be very different than from the diseases that we are dealing with.
Thank you so much. Okay, we have another question from Dr. Agustin Ruiz. This is for Dr. Hennica Michael. It's a very long question, so bear with me. We are now entering the stage where we are starting to tease apart systemic versus central inflammation, both of which might contribute to a mixed or overlapping signature, particularly in blood, but possibly also within the brain itself. This raises the questions of whether the inflammatory signals we detect peripherally truly reflect the CNS-specific processes or are confounded by broader systemic activity. I wonder whether there are strategic ways to disentangle this signal, perhaps through the integration of multi-omic approaches, cell-type deconvolution techniques, or spatial transcriptomics proteomics. It would be fascinating to hear your thoughts on how we might better resolve these overlapping layers of inflammation in neurodegenerative contexts. You're muted, Michael.
I'm sorry, that is a very difficult question, Agustin, and I'm trying to answer it to my best knowledge. I think it's likely that there are various influences. And of course, if you consider that there are pathological proteins, beta amyloid, and other neurodegenerative proteins are occurring in the peripheral blood, they are representing a danger-associated molecular pattern there as well. So if they hit peripheral immune cells, there is the possibility that they will initiate a similar reaction then in the brain. I believe that with very thorough multi-omics approaches, distinguishing, brain pathology and what happens in the brain from what happens in the periphery in the very same patient over time, and with many, with high numbers, we may learn what is what and where the different influences are. One way I would, kind of see that, which would be most straightforward, is to use brain imaging of inflammation using PET markers and developing PET markers of inflammation further, and correlating those with what happens in the brain and what happens in the periphery. We don't have a good idea when we look at blood samples if the inflammatory molecule we detect there comes from a single point in the brain or reflects a more wider immune reaction across a whole brain area or more. So we will only be able to unravel this if we have really an imaging marker for microglia activation. And maybe we need more than one marker for microglia activation, because we learn from transcriptomics and proteomics that there are many microglia subtypes in the very same brain. And we have to, to remember that this is a pathology that spreads throughout the brain, and by that spreading phenotype is supposedly having a temporal component that may affect microglia in the entorhinal cortex and hippocampus at a different time point to other brain areas.
Good. What do you think, Michael, about the current PET ligands for inflammation, microbial activation?
Well, it depends. If you globally want to label microglia by immunohistochemistry, I believe that you do use IBA1. If you have a PET marker, I still believe, if you refer to PET markers, I would still believe that you go with maybe PK11195. It's the best established one, but there are further markers in development. There has been a recent paper by Mastieu and colleagues, which showed a very interesting marker, which was showing activation of microglia cells concomitantly to A-beta deposition, which in my view makes sense because it's such a strong danger-associated molecular pattern to the neighboring immune cells. But as said, I think to really identify what microglia cells do, we may need more than one marker.
Cool. So to wrap up, I would like to ask a general question for all of you. I know that in brain banks, you receive patients in really late stages of cognitive decline. So what would be your ideal, brain that would you like to receive to study all the neurodegenerative disorders? Earlier brain, to have brains in all the continuum, or what kind of pathologies or brain would you like to use to advance this field?
Yeah, I could start. So I personally would go with a combination of, community-based longitudinal cohorts that are representing, cognitively normal without any memory impairment, but also having the full spectrum to end stage so that you can really assess the continuum as comprehensively as possible in the human brain.
I completely agree with this. So the brain collections that we have generally in brain banks do have patients who have very early stage disease and die from whatever other cause, not necessarily of the neurological disease. And especially the community-based or even the hospital-based cohorts can really identify these so-called incidental cases that we think that they are preclinical cases. So I think that it's just increasing the awareness of postmortem examinations can help to cover the whole spectrum.
What about you, Michael, with the field of immune activity and neuroinflammation? What kind of patient are you interested in? Brains are you interested in studying?
Well, I would be interested in the most important thing already has been said. I think we have a lot of cross-sectional information from the existing samples, and we need longitudinal samples. We need I would love to have brains actually from patients where we have clinical data, imaging data, blood, CSF, urine, fibroblasts, whatever you can get, but which have been really followed up over decades. And then I don't want to go so far, and because you said, what if you could wish, maybe I would even wish a brain biopsy to have fresh tissue. But that's probably one step too far. But I think we have to have more longitudinal information on what's going on, and ideally that would include, as I said, brain imaging, PET, MRI, atrophy measures on high Tesla brain volumetry, side by side with samples from CSF, side by side with plasma, and cognitive data. And this not only for 3-5 years of follow-up, I think we need that over decades, given the long trajectory of the disease.
Okay, I think we are close to the end. So thanks again on behalf of the organizing committee at Ace Alzheimer Center Barcelona to our three wonderful speakers, Dr. Flanagan, Dr. Kovacs, and Dr. Henneka, our audience and sponsors. Thank you all for being here today. So our next session of the Ace Global Research Summit is going to be about synucleinopathies, mechanisms, and implications for Alzheimer's disease on May 16th. So stay tuned and have a great weekend, everyone. So bye-bye.
Well, thanks for having us.
Thank you so much, the three of you.
Bye-bye.